Boyd, and I am presenting to you about my project that I've been working on since around November with my Denali, and it's interactions between human and plasmodium falciparum proteins using the yeast hybrid system. And there's a few key concepts and objectives for this lesson. Proteins in the cell interact with each other to carry out key functions, and then proteins interacting with each other give clues to the function. So if we want to understand how proteins work, we have to look at how they interact together. And then there are 5 steps to the yeast hybrid system: construction of the clay plasmid, construction of the baked plasmid, transformation of yeast cells, mating, and then selection. And we're going to study how molecules work and interact together at the foundation of activity. It contains work with genomes, biochemistry, genetics, and studies the interactions within a cell's own system. And we're going to see how we can understand proteins between how they interact together. And a few common procedures used to look at protein-protein interaction and molecules specifically are cross-linking, immunoprecipitation, and fractation by chromatography. And Marissa Bethany Boyd, who you guys know from Seattle Biomed, she used that procedure to look at malaria and human proteins. So that's looking at a pathogen and a host and how they react together. So the yeast hybrid system, the basis of the system is that if 2 proteins interact together, then they will survive. And that's simple, right? So if they don't interact, then the yeast cell dies, but if they do interact, then the yeast cell lives. We create the plasmids, and then we transform them into yeast cells. And we put it into a medium. And this medium lacks 3 amino acids. But the cool thing about this experiment is that the baked plasmid we talked about codes for the missing amino acid. And the prey plasmid we talked about codes for the other missing amino acid. So if these 2 interact, then it'll survive. And if it doesn't, they'll die. It allows the cell to grow in specific medias. And it is a useful tool because it identifies interactions between specific proteins by measuring simple growth or no growth readout. If a cell has the bait and clay plasmid, then it can go in the absence of both amino acids. In other words, unless a yeast cell has both plasmids, bait and prey, it will not grow in the media lacking both amino acids. And the thing about this is that you look at it, it's like, okay, that's cool. Maybe there was 3 reactions, however many. But actually, reading Marissa about paper, it's 46,000 interactions. Like, there's so many, and she's doing it on such a large scale. That's why things like cloning and gene delivery and other biotechnology techniques are really important. We're going to actually simulate the system. So I need 5 volunteers. Step 1 is creation of the first plasmid, which is the bait. So I'm going to need student ABB and student bait. Alright, so sorry. This is kind of a weird simulation, but you guys grab hands because you were interacting together. You were like moving a plasmid. So you're a plasmid. And then students. Sharon? Oh, sorry. Current student and AD student grab hands because you're creating the other plasmid. So you are gap or DBD fusing to the bait of the first plasmid. Oh, you are the gap or. And then you are the gap or AD fusing to the prey of the second plasmid. So now I'm going to need you to transform into yeast cells. So yeast is yeast cell A, so you guys get in there. And this is yeast cell alpha. Perfect. So you have DBD and bait. You are yeast cell mating type A. Student AD and prey are mating type alpha. So this is transformation. You guys have changed from plasmids to yeast cells. So now we're going to join the circles together to create to represent the primary method. Alright, so this is made from transforming into 1 cell to 2. Yes, so now we're going to react 2 different scenarios, and these are 2 different things that can happen. So, DBD will hand with UAS with the base student. Wait, that's you. Record. You join the group. Sorry. And so, student DBD, your UAS and the base student. So you are right down. Yes. Pray student, there's still one more. And then bait and pray will hand, both staying connected to either US or AD. So, right. So that's good. I'm good. The bait and pray are interacting. So you raise your paper. So this is now a yeast cell that is interacting, and you guys are alive. So this is a really good situation. The bait and the prey have interacted. That's you two holding hands, and so you guys can look. Now, everyone got the answers here. So you were alive. That was situation 1. But now we're going to go over situation 2. So DBD holds hands with UAS student and a bait student. So you guys can go back to that. Prey student holds hands with the ED student. So the bait student and the prey student does not hold hands. And the US student, that's you, you're the recorder, you do not raise your paper because you guys are dead. There's no way that the bait can interact with the prey. The prey can interact with the bait, and so you guys can't exist as a yeast cell. And so, the key message of the first scenario is when the bait and the prey interact. HIS is activated, that's you, and the yeast is able to grow in the selected bait. But when they're not, when the bait and prey do not interact, HIS is not activated, and the yeast is unable to grow in the selected bait. So, that you guys can again. What we just talked about and the 5 steps you just wrote about can be used to study pathogen and host. And that's like, again, notes repetition, but that's a really key molecular biology technique that you can put anything into those bait and prey positions and see how they interact and what they do. And is the yeast cell they create going to live, or is it going to not in the selected medium? And so it's only looking at part of the cycle, and it's only looking at part of the interaction. And so that's on a very small level. So it might not make sense why it applies to curing malaria or looking at it analytically, but it really is a very important process, and what she did was a lot of work. So the yeast to hybrid system is used to understand how plasmodium proteins at the interface between the parasite and the host cell, cytoplasm could be interacting with human proteins, such as a protein. So she used her proteins and key falciparum as her bait. So I have another question for you. The yeast to hybrid system can be used to research the pathogen and host interaction. So, applying what you know about malaria and parasites, how do you think it could fit into using the yeast to hybrid system?